Welcome to Biotanime.
Endospore. An endospore is a dormant, tough, and non-reproductive structure produced by certain bacteria from the Firmicute phylum. Endospore formation is usually triggered by lack of nutrients, and usually occurs in gram-positive bacteria like Bacillus and Clostridium. The process of forming endospores is called sporulation. Endospores enable bacteria to lie dormant for extended periods, even centuries. When the environment becomes more favorable, the endospore can reactivate itself to the vegetative state. The endospore consists of the bacterium's DNA and part of its cytoplasm, surrounded by a very tough outer coating. Endospores can survive without nutrients. They are resistant to ultraviolet radiation, desiccation, high temperature, extreme freezing and chemical disinfectants. They are commonly found in soil and water. The stages in endospore formation are: Stage 0, vegetative cells with 2 chromosomes at the end of exponential growth. Stage 1, formation of axial chromatin filament and excretion of exoenzymes, including proteases. Stage 2, phosphorseptum formation and segregation of nuclear material into 2 compartments. Stage 3, spore protoplast formation and elevation of tricarboxylic acid and glyoxylate cycle enzyme levels. Stage 4, cortex formation and refractile appearance of spore. Stage 5, spore coat protein formation. This stage it is called as phosphor. Stage 6, spore maturation, modification of cortical peptidoglycan, uptake of dipicolinic acid, a unique endospore product, and calcium, and development of resistance to heat and organic solvents. And stage 7, final maturation and liberation of endospores from mother cells. Newly formed endospores appear as round, highly refractile cells within the vegetative cell wall, or sporangium. The spore protoplast, or core, contains a complete nucleus, ribosomes, and energy-generating components that are enclosed within a modified cytoplasmic membrane. The peptidoglycan spore wall surrounds the spore membrane. On germination, this wall becomes the vegetative cell wall. Surrounding the spore wall is a thick cortex that contains an unusual type of peptidoglycan, which is rapidly released on germination. A spore coat of keratin-like protein encases the spore contained within a membrane called exosporium. A special stain technique called a molar stain is used to stain endospore. This allows the endospore to show up as red, while the rest of the cell stains blue. Another staining technique for endospores is the schaeffer-Fulton stain, which stains endospores green and bacterial bodies red. Endospores can be destroyed by burning or by autoclaving. Endospores are able to survive boiling at 100 degrees Celsius for hours, although the longer the number of hours the fewer that will survive. An indirect way to destroy them is to place them in an environment that reactivates them to their vegetative state. They will germinate within a day or two with the right environmental conditions, and then the vegetative cells can be straightforwardly destroyed. This indirect method is called tyndallization. Geobacillus stereodermophilus endospores are used as biological indicators when an autoclave is used in sterilization procedures.
Thank you. Please like, subscribe and share. See you again in a new topic.